Thank you, David, for that wonderful talk. The next speaker is Marcia Haigis, and Marcia received her PhD from the University of Wisconsin in 2002, and as Tyler mentioned, did her postdoctoral training at MIT with Lenny Guarente. In 2006, she took a position as an assistant professor of pathology at Harvard Medical School and is also a member of the Paul F. Glenn Laboratories for Biological Mechanisms of Aging. Her work has contributed to showing that the mitochondrial sirtuins regulate metabolism in aging, and she's going to tell you today about the role of these proteins in tumor cell metabolism. Marcia, it's a pleasure to have you here.
So I want to thank the organizers for inviting me today. I remember during my time at MIT as a postdoc in Lenny Guarente's lab that this symposium really is such a highlight for the community. So thank you. Can you advance to the next slide, please? So my lab is interested in the central question of how does mitochondrial metabolism contribute to aging and age-related disease? And what we're interested in is in understanding how pathways of aging, such as sirtuins, contribute to changes in mitochondrial metabolism, mitochondrial signaling, during different diseases of aging. And a large proportion of my lab studies diabetes and metabolic syndromes and disorders. But we've become growingly interested in the area of cancer metabolism because, as you hear today, there's such an overlap between mitochondrial metabolic networks in the area of diabetes and how cells use energy to regulate the relationship between anaplerosis and catabolic pathways. So sirtuins are highly conserved from bacteria to humans, and in mammals there are 7 sirtuins called Sirti1 through 7. They're first identified by a conserved sirtuin core domain in yeast Sir2. Sirtuins have, thank you. And so sirtuins have a highly conserved core domain that contains their catalytic activity, and they have a divergent flanking sequences that likely help to dictate their substrate specificity as well as different subcellular localizations. Sirtuins have a really interesting enzymatic activity. They function as NAD-dependent deacetylases or ADP ribosyl transferases. Their absolute reliance on NAD makes them a really interesting metabolic sensor. So their activity is coupled to the metabolic state of the cell. 3 sirtuins are localized in the mitochondria, Sirti3, 4, and 5. And this localization is particularly intriguing to us because recent proteomic analyses of lysine acetylation has shown surprisingly that approximately 15% of mitochondrial proteins can be modified by acetylation, suggesting that their deacetylation and the levels of this modification can be modified by mitochondrial sirtuins. Moreover, the substrates or the proteins that are acetylated are themselves in central players of mitochondrial metabolism and could potentially be regulators of how mitochondrials use fuel. So for instance, components of fat oxidation are heavily acetylated. Components of the TCA cycle, electron transport chain subunits, as well as amino acid and metabolic enzymes are also acetylated. And intriguingly, although mitochondrial sirtuins, in comparison to Sirti1, which is the mammalian ortholog of Sir2, which is the most heavily studied sirtuin, mitochondrial sirtuins are not as well studied But the few studies that have come out recently have shown that there's a really elegant coordination of how sirtuins are working together in the mitochondria to regulate how cells use fuels. So for instance, both SIRT4 and SIRT3 bind to an enzyme called glutamate dehydrogenase. When I was a postdoc in Lenny Garante's lab, we showed that SIRT4 binds to GDH and represses its enzymatic activity. SIRT3 also stimulates fatty acid oxidation during times of nutrient deprivation to promote fat oxidation in the mitochondria. SIRT3 also binds to multiple subunits of the electron transport chain, including subunits in complexes 1, complex 2 in SDH, as well as complex 3. And so there is a coordination of mitochondrial metabolism by sirtuins, how individual sirtuins work together to maintain energy homeostasis in cells under different energy conditions, under different stress conditions, is still not well understood. Among the mitochondrial sirtuins, SIRT3 is the most studied sirtuin. And so shown here is a list of its target proteins, often interacting proteins that are themselves acetylated, and the pathways that are regulated. And so, for instance, SIRT3 binds to LCAD and deacetylates it, stimulating fatty acid oxidation. So the net effect of SIRT3 activation is in stimulation of mitochondrial oxidative metabolism. I also want to point out that SIRT3 has a newly emerging role in the maintenance of redox balance and ROS clearance in the cell. So it's very interesting that in addition to making the electron transport chain more efficient, SIRT3 also helps to promote the clearance of free radicals by, for instance, deacetylating SOD2. And so because of its wide variety of mitochondrial substrates, we became very interested in a general question of how does SIRT3 function or loss of function impact general cellular metabolism. And so this is a project really driven by a talented graduate student in the lab, Lydia Finley, and she undertook a metabolomic approach in collaboration with Clary Cleach here at the Broad Institute. And what we did was we harvested metabolites from wild-type or knockout littermate MAFs, and we analyzed polar metabolites by LCMS. And what we found was a complete metabolic shift in the redistribution or the ratios of glycolytic metabolites and TCA cycle metabolites. So metabolites of glycolysis that were measurable were increased in SIRT3 knockout cells, while many metabolites in the TCA cycle were decreased. And for instance, if you look at the data, intracellular glucose levels were decreased. Moreover, a metabolite in the pentose phosphate pathway was increased with loss of SIRT3. Now, these are snapshots, and so to dynamically measure changes of fuel utilization by loss of SIRT3, we measured glucose uptake and found that, indeed, there was a dramatic increase in glucose uptake and usage by the cells, and also an increase in lactic acid secretion. And this effect is often referred to as the Warburg effect, as Matt nicely introduced earlier today. And so I don't have to go into the details, but in a quiescent state, most of the metabolized glucose and pyruvate is utilized by mitochondria to maintain energy in the cell, whereas during a rapidly proliferating state, such as a tumor cell, glucose is, the pyruvate from glucose is metabolized to lactate. Furthermore, glucose metabolites are also used for other pathways for building biomass, such as the pentose phosphate pathway. So about this time we were generating this data, an interesting paper came out from David Geis's lab demonstrating that SIRT3 is a tumor suppressor. So SIRT3 knockout mice are completely viable. They look identical to littermate controls. They have normal development, and there's no apparent detectable phenotype unless you start to probe individual processes under the exactly right condition. But with age, so about at 12 to 18 months, 50% of SIRT3 knockout mice spontaneously develop mammary tumors, and the mechanism that was responsible for this involved ROS, cellular reactive oxygen species, that increased in SIRT3 knockout cells, promoting genomic instability and tumorigenesis. We had a different idea for this. We had a different question. We wanted to know, because SIRT3 functions primarily in the mitochondria to bind and regulate so many metabolic enzymes, and because we discovered that SIRT3 results or controls this global cellular reprogramming from mitochondrial metabolism to glycolytic metabolism, could SIRT3 also contribute to tumorigenesis via metabolic reprogramming? And of course, this could give many different advantages to tumor cells, including rapid ATP generation, survival during or under hypoxic conditions, as well as this biosynthetic growth advantage. And so we first tested this idea by simply measuring their proliferation rate of SIRT3 wild-type or knockout MAFs under high glucose conditions. And we saw that indeed, loss of SIRT3 results in cells that grow faster, that divide faster. And this is entirely dependent on glucose. So when we take those same cells and measure their proliferation in media containing no glucose, now containing galactose, we can see that they have an identical growth rate. Moreover, this is true in vivo. So we've done FDG PET imaging of glucose uptake, and we can see that SIRT3 knockout mice in the brown adipose tissue uptake more glucose than control animals. And when we analyzed the pathways involved, we were quite surprised. So we performed microarray analysis of wild type compared to littermate knockout brown adipose tissue. Because SIRT3 is a mitochondrial metabolic enzyme, we thought that we kind of expected a number of mitochondrial pathways to jump out, maybe compensating, perhaps involved in mitobiogenesis, et cetera. But instead what we found was that SIRT3 loss upregulated a number of tumorigenic pathways, such as RAS signatures, MYC signatures, as well as hypoxic signatures that were dramatically upregulated during SIRT3 loss. And this really piqued our attention. So we analyzed how does hypoxia affect, how does SIRT3 affect the hypoxic response? And so during normoxia, we can see that SIRT3 knockout cells start out with higher levels of glycolytic intermediates. During hypoxia, wild-type cells increase the levels of glycolytic intermediates, and this response is even more dramatically upregulated in SIRT3 knockout cells. And so what's the mechanism involved? And so we became very interested in a master regulator of hypoxia called HIF-1 alpha. And during hypoxia, HIF-1 alpha is stabilized and forms a dimer with HIF-beta. And that translocates to the nucleus to drive the expression of genes that allow cells to adapt to low oxygen conditions. So for instance, HIF-1 alpha activity drives the expression of GLUT1 and H-hexokinase 2 to increase glycolytic metabolism. Now, during normoxic conditions, HIF is greatly destabilized, HIF-1 alpha is greatly destabilized, because during normoxia, PHDs are activated, and they hydroxylate residues on HIF-1 alpha that render it a very good substrate for VHL activity. And VHL is a ubiquitin ligase that then targets HIF-1 alpha for proteolytic degradation. And so HIF-1 alpha activity really, by driving changes in gene expression, helps cells decide whether or not to use oxidative phosphorylation or glycolytic metabolism, thereby helping the cell deal with low oxygen conditions and kind of deal with maintaining ROS or detriments of low oxygen. And so because HIF-1 alpha maintains this shift, we wanted to know, could SIRT3 regulate HIF-1 alpha, thereby maintaining the balance between glycolytic metabolism and oxidative phosphorylation? And so what we did was we measured HIF levels in wild-type or knockout mouse, and we found that indeed during 0, 6 hours, 12 hours, or 24 hours of 1% oxygen, we saw that there was increased HIF-1 alpha stabilization. This is reflective also of HIF-1 alpha activity because a number of HIF target genes are also upregulated in SIRT3 knockout cells under normoxia or hypoxia. Moreover, in SIRT3 overexpressing cells, and here shown are 293 T cells, SIRT3 overexpression destabilizes HIF and downregulates expression of HIF target genes. Now, initially, when we got the results that SIRT3 loss upregulates glycolytic metabolism, we also tested whether or not this upregulation of glycolytic metabolism was merely compensation for defects in mitochondrial function. First of all, SIRT3 loss causes very subtle defects in respiration and mitochondrial function, and it doesn't depend on mitochondrial respiration or fat oxidation defects. But when you knock down HIF-1 alpha, now you see that there is no difference in GLUT1 expression or lactic acid secretion with SIRT3 loss. So HIF is absolutely essential for metabolic reprogramming by SIRT3. So how is it stabilized in SIRT3 knockout cells? This is something that really took us a long time to work out, and we were very interested in this pathway and how PHT function was regulated. And so first, to probe whether or not PHT was involved, we looked at HIF-1 alpha hydroxylation levels by using the presence or absence of MG132, which is an inhibitor of the proteasome, or DMOG to inhibit PHT function. And basically, you could see that in SIRT3 knockout cells, there was a dramatic decrease in the level of HIF hydroxylation, suggesting that the regulation was upstream. And so SIRT3 is known to regulate ROS, and this is shown in the last just year or two by multiple studies. So first, SIRT3 can regulate SOD directly by deacetylation of multiple lysine residues. SIRT3 also regulates complex 1, complex 3 activity, and so thereby can affect the efficiency of electron transport and ROS leakage from those complexes. So we tested the model that SIRT3 loss regulated ROS, which would then regulate PHD activity. And indeed, hypoxia induces ROS levels, and we saw that in normal cells. And with SIRT3 loss, we saw a greater induction of ROS level. To test the hypothesis that ROS was indeed responsible for the stability of HIF in SIRT3 knockout cells, we rescued that by the addition of N-acetylcysteine antioxidant. And we found that indeed, NAC addition normalized glycolytic gene expression in knockout cells, as well as normalized the growth rate in knockout cells. And so in summary, we found that SIRT3 can regulate HIF-1 alpha stability and activity, thereby mediating a glycolytic switch. So when you have SIRT3, you repress HIF-1 alpha, and you promote mitochondrial metabolism through gene expression, rapidly shutting down glycolytic pathways. And when you lose SIRT3, you shut down mitochondrial metabolism, and you promote glycolytic gene expression. This is actually an additional layer on how SIRT3 works, because SIRT3 primarily is localized in mitochondria and primarily activates mitochondrial oxidation by its deacetylase activity. So this is a really interesting kind of dual level, additional level of how SIRT3 regulates cellular metabolism. First, by its deacetylase activity directly on mitochondrial substrates in metabolic pathways. Second, by regulating HIF-1 alpha activity and stability to drive changes in glycolytic gene expression. So the big question we wanted to ask next is, does this pathway, does SIRT3 have anything to do with tumor cell metabolism? And so previously it was shown by David Geis's lab that SIRT3 knockout cells grow larger tumors in xenografts of tumor formation. And we repeated this result, and we further analyzed these tumors for differences in glycolytic gene expression to see if indeed SIRT3 knockout tumors had an altered metabolism. And we did see that there was increased glycolytic gene expression, and you can see increased GLUT1 IHC staining here. Moreover, when we used the TumorScape database to analyze SIRT3 copy number across human tumors, we saw something really striking, which was that a large number of human tumors, about 20% in all, had a SIRT3 deletion. Strikingly, breast tumors and ovarian tumors had a 40% deletion of one copy of SIRT3, including a focal deletion. So this is a SIRT3 copy number, SIRT4 and 5 as controls, and P53. And the blue shows the level of deletion across different tumor types. By IHC analysis in a blind study by Julie Terura-Feldstein at Sloan Kettering, she saw an even striking difference in the SIRT3 expression between normal and tumor breast cancer cells, in that about 85% of human breast cancers showed reduced SIRT3 expression. Moreover, we still are trying to understand and probe how important the relationship between SIRT3 and HIF-1 alpha is in human cancer, and that's an area we're starting to investigate now. But in these human breast tumors, what we do know is that there is a strong inverse correlation between SIRT3 and glycolytic gene expression. So for instance, shown here, the higher the SIRT3 expression in the human tumor, the lower the GLUT1 expression, and the reverse is true. So next we wanted to know, is SIRT3 loss of function relevant to human cancers? What's the effect of SIRT3 overexpression on the metabolism of human cancer? I showed detailed mechanistic studies of how SIRT3 regulates HIF-1 alpha in 293 T cells or in knockout MEFs, but what about tumor cells? And so what we did was we took a panel of breast cancer cells, shown here are KAMA1 cells or T47D cells, and we overexpressed SIRT3. These are cells with low copy number of SIRT3. And when you overexpress SIRT3, you can see under normoxia, or shown here during hypoxia, SIRT3 decreases glycolytic gene expression or gene expression of transcripts involved in glucose metabolism in both cases. Moreover, SIRT3 overexpression also results in lower levels of HIF-1 alpha during hypoxia. As you would expect from changes in glycolytic gene expression, SIRT3 overexpression in a panel of breast cancer cells also represses glucose uptake and lactic acid secretion from the cells, suggesting that, again, in this case, as well as in the MEFs and other cell lines we've looked at, SIRT3 overexpression represses glycolytic metabolism. So does SIRT3 affect the growth of breast cancer cells? Not in every cell we've looked at, but in KAMA1 cells, SIRT3 overexpression represses the growth of KAMA1 cells in a glucose-dependent manner. And interestingly, consistently, in studies we do in KAMA1 cells in the absence of glucose, we see that SIRT3 overexpressing cells actually grow a little bit faster. And so we've identified a novel regulation of how SIRT3 controls metabolism outside of its direct role as a mitochondrial deacetylase. So SIRT3 regulates ROS levels, and that ROS acts as a signal to regulate HIF-1 stability, thereby helping a cell decide whether or not to actively boost mitochondrial metabolism through repression of HIF and activation of SIRT3 deacetylation, or boost glycolytic metabolism. We also want to know, are there other more direct molecular targets of SIRT3 that regulate HIF? How much do these metabolic reprogramming networks contribute to tumorigenesis? I showed earlier that David Geis's lab showed that this increased ROS and SIRT3 knockout cells contributes to genomic instability. So what's the interplay between genomic instability and metabolic contributions to tumorigenesis? And does SIRT3 activity or level also correlate with prognosis, tumor grade, or therapeutic outcome in patients? And interestingly, a really elegant study performed by Eric Bell in Lenny Garante's lab also showed that SIRT3 regulates HIF-1 stability. And so just in the last couple minutes of the talk, I want to switch gears and talk about a different sirtuin. So I focused most of my talk on how a mitochondrial sirtuin, SIRT3, regulates glycolytic arm of cellular metabolism through signaling. From previous work that we've done, we know that SIRT4 represses glutamate dehydrogenase. Interestingly, tumor cells use glutamine, and many tumor cells use more glutamine than normal cells. And that is very important for biosynthesis of materials and also for energy production in tumor cells. So we examined how SIRT4 regulates glutamine utilization in a panel of different human cell lines, including tumor cells. And in most cells, we look at SIRT4 overexpression, but not SIRT4 overexpression of a catalytic mutant, results in a repression of glutamine utilization by cells, results in a repression of ammonia secretion from cells. Interestingly, SIRT4 whole body knockout mice spontaneously develop tumors. In collaboration with Zhuzha Ding, we've looked at two separate, independently generated SIRT4 knockout mouse lines and found that in each case, there was an increase in lung tumor formation spontaneously with age. So we see this phenotype most clearly at about 18 to 22 months. And so clearly now we're looking at what's the contribution of glutamine metabolism or genomic instability to these phenotypes as well. Unlike SIRT3, I just want to point out, SIRT4 seems to have a very modest effect on glycolytic metabolism, so this is probably independent of the canonical Warburg effect. So in summary, I've shown you that SIRT3 regulates glycolysis in addition to mitochondrial metabolism by regulation of HIF-1 stability. SIRT3 regulates both tumor cell metabolism and genomic instability, really acting as a two-pronged or a two-hit regulator. SIRT4 may also have a role as a tumor suppressor, and I think by looking at this pathway of proteins, we can uncover novel roles for regulators of mitochondrial metabolism and how that interacts with genomic fidelity. So the HIF-1 SIRT3 work was really spearheaded by Lydia Finley. Jae-Won Lee, a medical student in the lab, contributed to this work greatly. Singmin Jung is a very talented postdoc in the work who's really driven all of the SIRT4 tumorigenesis and glutamine addiction story. We have a number of other collaborators. So our tumor work was greatly assisted by Pierpello Pandolfi, metabolomics work and bioinformatics work by Clary Cleach and Jang-Wang Zhen. We used Sirtuin knockout mice generated by Fred Alt's lab. We're collaborating with Zhuzha Deng and Alec Kimmelman to examine the role of SIRT4 in metabolism. Thank you.
Thank you, Marcia.